Having gone over the cell cycle in the last video, we can explore the stages of mitosis and cytokinesis. During G2, before mitosis begins, the cell has a well-defined nuclear envelope that surrounds the nucleus. There is one or more nucleoli present. There are two centrosomes composed of centrioles in animal cells that form an aster complex. Plant cells have centrosomes but no centrioles and can still form the spindle structure. Recall that centrosomes are the microtubule organizing center. The genetic material is in the form of chromatin, uncondensed. The first stage of mitosis is prophase. In prophase, the nucleoli disappear. The chromatin condenses into chromosomes using histone proteins. The chromosomes are composed of two sister chromatids attached at the centromere. Remember that DNA was duplicated during S phase, so there is double the amount of genetic material. So, two sister chromatids make up one chromosome. The mitotic spindle arises, which consists of microtubules coming from the centrosomes. Then the centrosomes begin to move apart. During prometaphase, the nuclear envelope disintegrates, allowing the microtubules to interact with the chromosomes. The mitotic spindles move to opposite sides of the cell, and the microtubules extend. Some microtubules attach to the kinetochores of chromosomes. These microtubules are called kinetochore microtubules. Non-kinetochore microtubules do not attach to chromosomes and end up overlapping with microtubules from the other side of the cell. This elongates the cell. Microtubules lengthen or shorten by adding or removing tubulin protein. Next comes metaphase. In metaphase, the centrosomes are on opposite ends of the cell. The chromosomes are aligned on the metaphase plate. This marks the metaphase checkpoint. Both the non-kinetochore and kinetochore microtubules are referred to as the spindle. In anaphase, the sister chromatids split at the centromere and become mature chromosomes. They move to opposite ends of the cell. The chromosomes form a V shape. Kinetochore microtubules shorten as the chromosomes move away from the center, and non-kinetochore microtubules move farther apart, elongating the cell. At the end of anaphase, the two ends of the cell have identical chromosomes. In the final stage of mitosis, telophase, chromosomes decondense into chromatin. The non-kinetochore microtubules move further apart, elongating the cell. The nuclear envelope reforms. Nucleoli reappear. The formation of two identical nuclei marks the end of mitosis. Then cytokinesis begins, where two new daughter cells arise. In animals, cytokinesis occurs through the formation of a cleavage furrow. A shallow groove forms on the cell surface, and a contractile ring composed of actin microfilaments contract until the parent cell pinches off into two new daughter cells. Cytokinesis in plant cells occurs a bit differently due to the cell wall. Vesicles form on the metaphase plate and then merge to form a cell plate. Cellulose is then deposited, and a new cell wall forms between the two new daughter cells. That's it for this lesson. In the next video, we will go over the process of meiosis. See you then. For more bio content, hit subscribe.